Hello viewers, welcome to our channel Biological Concepts. In today's video, we will discuss some important questions on chromosomes. Hello viewers, welcome to our channel Biological Concepts. In today's video, we will discuss some important questions on chromosome. Some brief notes regarding the questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question. Two sister chromatids are attached with option 1 chromomere, option 2 chromonema, option 3 centromere and option 4 is centrosome. And the correct answer is option 3 centromere. Now let's discuss about this centromere. Centromere links a pair of sister chromatids together during cell division. This constricted regions of chromosome connects the sister chromatids, creating short arm P and long arm Q on the chromatids. During mitosis, the spindle fibers attach to the centromere via the kinetochore. Next question. Chromosome can be stained with option 1 acetocarmine, option 2 safranin, option 3 fulgen and option 4 is methylene blue. And the correct answer is option 1 acetocarmine. The stain, that is acetocarmine dye, is used for dyeing the chromosome. This stain is a DNA specific stain and is used when the study of different mitotic stages is required. To stain the chromosome, first acetocarmine dye is prepared using the carmine. This carmine is a basic dye and which is obtained from the offspring insect. Next one. Male human chromosome is represented by the sex chromosome option 1 XX, option 2 XY, option 3 XO and option 4 is YY. And the correct answer is option 2, that is XY. Whereas the XX represents the female human chromosome, the XO represents the Turner syndrome and XXY represents for Klinefelter syndrome. The amino acids that make up the histone proteins are option 1 lysine and arginine, option 2 lysine and glycine, option 3 methionine and arginine and option 4 is threonine and arginine. And the correct answer is option 1 lysine and arginine. The basic amino acids that make up the histone proteins are called is arginine and lysine. They are basics as they have the positive charge on their side chain at physiological pH. Because the presence of these amino acids, that is lysine and arginine, the histone proteins are positive in charge. This positive charge histone proteins helps in the coiling of DNA. These histones can be divided into two groups based on the amino acid content. The H1, H2A, H2B are lysine rich, whereas the H3 and H4 are arginine rich histones. Next one. Nuclear organizer region contains option 1 rRNA, that is ribosomal RNA, option 2 mRNA, that is messenger RNA, option 3 tRNA, that is transferred RNA, option 4 is all of the above. And the correct answer is option 1 rRNA, that is ribosomal RNA. The nuclear organizer region are chromosomal region which is crucial for the formation of the nucleus. The nuclear organizer region is the specific region at which nucleolus is attached to the chromatin fiber. This region codes for 5.8S, 18S and 28S rRNA. It was first discovered by Barbara McClintock in 1934 in GMAs. Next question. Satellite DNA present in option 1 euchromatin, Option 2 heterochromatin. Option 3 both 1 and 2. And option 4 none of the above. And the correct answer is option 2 heterochromatin. Now let's discuss about what is satellite DNA. This satellite DNA is the primary structural component of heterochromatin, which is found in functioning centromere. The satellite DNA is made up of vast clusters of non-coding, tandemly repeating DNA. Next question. Nucleolar organizer region contains genes which codes for option 1 5.8 is rRNA, option 2 18 is rRNA, option 3 28 is rRNA, option 4 all of the above. And the correct answer is all of the above, that is 5.8 is rRNA, 18 is rRNA, 28 is rRNA. We have already discussed that this nuclear organizer region coding for all these rRNA. It is the chromosomal regions which is crucial for the formation of nucleolus, and this regions codes for 5.8 is 18 is and 28 is rRNA. Next question. What is the function of centromere in chromosome? Option 1 formation of spindle fiber, option 2 movement of chromosome, and option 3 division of centrosome. Option 4 both 1 and 2. Option 5 is both 2 and 3. And the correct answer is option 4, that is both 1 and 2. That is the function of centromere in chromosome is the formation of spindle fibers as well as in the movement of the chromosome. Now let's discuss about what is this centromere and what is its function in details. Centromere are the point of attachment of the kinetochore. The main function include attachment of sister chromatids, and it is the site for the attachment of spindle fiber. The centromere help in proper alignment and segregation of the chromosome during the process of cell division in eukaryotic cells. The centromeres plays an important part in the production of new cell. When the chromosomes are copied, the centromere serve as the binding site for the two new replicated chromosomes, which are known as the sister chromatid. The centromeres are also involved in the sister chromatid adhesion and separation, chromosome movement, microtubule attachment, and the establishment of heterochromatin and mitotic checkpoint control. These are all at the function of the centromere. The centromere also function as sophisticated signal processing center, which are involved in the regulation of cell cycle progression. Next question. Nucleolar organizer region is separated from the rest of the chromosome by option 1 is primary constriction, option 2 secondary constriction, option 3 chromomere, and option 4 is centromere. And the correct answer is option 2 secondary constriction. Now let's discuss about this secondary constriction. Secondary constriction are constricted or the narrow region. The secondary constriction is a constricted or narrow region at any point of the chromosomes other than the of centromere. The centromere is known as the primary constriction. The difference between these two constrictions can be noticed during the anaphase. These secondary constrictions are useful in identifying chromosome from a set. Some parts of this constriction indicate the sites of nucleolus formation and are called the nucleolar organizing region, as it indicates the sites for nucleolus formation, so it is known as the nucleolar organizer region. Okay. Next one is polytenic chromosome discovered by option 1 Biani, option 2 Hertz and Bower, option 3 Wiseman, option 4 is Watson and Crick, and the correct answer is option 1, that is Biani. Now What is this polygyny chromosome? These polygyny chromosomes are large chromosomes containing thousands of DNA strands. And these polygyny chromosomes provide high level of functions in certain tissues such as salivary glands of insects. So this polygyny chromosome can be found in the salivary glands of insects. Next question. Positive charge of histone protein is due to option 1 lysine and arginine, option 2 proline and lysine, option 3 fluorine and methionine, option 4 histidine and methionine. And this correct answer is option 1, that is lysine and arginine. These amino acids have positive charge on their side chains at physiological pH. Okay. These positive charge histone proteins help in coiling of DNA. Due to the presence of this lysine and arginine, the histone proteins are positively charged. And these positively charged histone proteins helps in coiling of the DNA. Next question. Nucleosome core particle is, you have to fill in the blanks, that pair of DNA. The option 1 is 146 base pairs, option 2 246 base pairs, option 3 300 base pairs, and option 4 is 270 base pairs. And the correct answer is option 1, 146 base pairs. The nucleosome core particle consists of about 146 base pairs of DNA that is wrapped in 1.67 left-handed spherical turns around the histone octamer. This histone octamer consisting of 2 copies each of the core histones, that is H2A, H2B, H3 and H4. The adjacent nucleosomes are joined by stretch of free DNA, which is termed as linked DNA, which varies from 10 to 80 base pairs length depending on the species or tissue type. Next question. Number of autosomes in human are option 1 22 pairs, option 2 23 pairs, option 3 2 pairs, and option 4 is 10 pairs. And the correct answer is option 1, that is 22 pairs. Humans have 23 pairs of chromosomes, that is 46 in all. Among these 23 pairs of chromosome, 22 pairs, that is 44 are autosomes, and rest 2, that is the 1 pair, are sex chromosomes. This sex chromosome is also known as the heterosome. The sex chromosomes is represented in female as XX, whereas in male it is represented as XY. Next question. Histone protein not present in the core of nucleosome. Option 1 H1, option 2 H2A, option 3 H2B, and option 4 is H4. The correct answer is option 1 H1. This H1 histone protein is not present in the core of nucleosome. Each nucleosome consists of little less than 2 turns of DNA, which is wrapped around a set of 8 proteins called histones. These are known as histone octamer. Each histone octamer is composed of 2 copies each of the histone proteins, that is H2A, H2B, H3 and H4. Whereas the linker histones is known as H1 and its isoform is believed in the chromatin compaction and sit at the base of the nucleosome near the DNA entry and exit binding the linker region of the DNA. The 5 major families of histone proteins exist, which are H1 or H5, H2A, H2B, H3 and H4. Histone H2A, H2B, H3, H4 are known as the core or nucleosome histone, whereas the H1 or H5 are known as the linker histone. So, in core histone, H1 or H5 is not present. It only consists of H2A, H2B, H3 and H4. So, these are all about the questions on chromosomes. If you like this video, please like, share and subscribe to this channel. Thank you.